Okay, sorry the video was cut up. So we're talking about 6 histidine tag. Instead of a GST tag, you can add another tag called hexahistidine. Hexa means 6. So you can add extra 6 histidine. Histidines are what? Amino acids. So 6 histidines in a row, they will develop a unique structure. So those are the 6 histidine amino acid that can bind with nickel ion. So nickel ion will be the affinity chromatography resin, and then you can purify your protein that has a 6 histidine tag over the nickel ion column. So this is another tag protein available, which is a much smaller protein because only like 6 histidine, very small. Okay, so polyhistidine tag offers advantages for your protein purification because it's a very small tag made up with only 6 histidine only. It doesn't render the structure of your protein a lot, so which is a strong advantage. So it can be added on the N terminus or C terminus. Once it's added, it can bind with the nickel ions strongly. And then how do you elute it? You add the structure that has similar formation like 6 histidine. We call it imidazole. Imidazole is added. Again, it's based on the computation. Then your protein will be eluted. That's how it works. Okay. So example is given: marker band, and probably it's a cell lysate element. It means cell lysate, entire cellular protein is loaded as an example. Flow through, FT means flow through. So probably you did the purification of a nickel ion column, and before maybe the entire cell lysate was added, you added a little bit of that sample. And flow through meaning sample was bound and then unbound molecules were captured. That's flow through. And the washing fractions, okay, you do a lot of washing. Remember, chromatography run, sample binding, washing, and elution. Okay, so it seems like that is the target protein, which is prominent in your cell lysate, right? But it's not there on the flow through, and not much there in the washing. That is not the same protein as this. Okay, so only elution has a lot of this. Okay. So it's a batch binding, it says, and spin column. So maybe another column they used it to capture a pure. Probably that's what it shows. Okay, pure resin, another type. So very pure protein is purified. That's what it shows. In the cloning strategy, of course, there's the 6 histidine tag. It can be added on the C terminus or N terminus, depends on how you clone your gene. If it's cloned like that, it's probably added on the C terminus of your protein. Or if it's added the other way around, it's going to be added on the N terminus. It doesn't matter whichever it goes. Okay, so those are the 6 codons code for 6 histidine. This could be a N terminus tag. If it's added here, the 6 histidine on the C terminus tag. Okay, either way is fine. And elution strategy, again, you add competitor, which is imidazole, in this case imidazole. Okay, so that has the strong that structure which will compete against the nickel ion. Okay, so you can elute a tag protein. Same thing over. So sample loading, binding, washing, and elution is taking place. And then the question is, how are you going to remove imidazole that is not needed? And you can remove imidazole from your protein. Let's say the red balls are the imidazole that you don't want, or salt. They can be removed through desalting column. What is a desalting column? That's a size exclusion chromatography. Remember how that works? In the resin balls, there are small channels. So small molecules will run through the small channels in the resin ball, but the large molecules run through the column much faster. So that's how you can purify your target protein from imidazole. So you can remove imidazole efficiently. That's what it shows. Another example, this shows probably washing, elution, or something like that. Start up elution. I guess the elution is using it's probably conductivity, it's a salt, and you see different types of proteins are purified together here. Okay, so you can take a look at what it means. Column is nickel column, histidine trap, okay, and then sample is N-terminus 6 histidine tagged E. coli fusion protein, okay, flow through 0.1 to 1 ml per minute, sample loading, and binding buffer 20 millimole tris, 0.5 mole sodium chloride plus 5 millimole imidazole. Binding is done with the presence of small imidazole. Washing, same condition, but you have more imidazole there, 20 millimole imidazole. Refolding, again, same condition, okay, barium capital ethanol, and then refolding gradient. So what it means is, your protein is purified under the denaturing condition. That's what it means, starting at this condition. So, and then over the column, you can slowly refold your protein, okay, by removing probably salt or something like that. And then while that's happening, you can also purify your target protein. So at the end, elution buffer contains 500 millimole imidazole. So you can have it come off. So same other conditions are about the same. So the difference is here, when you do the binding, you have 6 molar guanidine hydrochloride. That's a strong denaturant. So the binding is done, seems like under the denaturing condition, okay, and then during the washing, you're removing that 6 molar guanidine. Okay, so denatured condition and washing it, your protein is slowly refolded. That's what it means. Okay, while it's refolded, other proteins are removed, and then you can capture your protein with the strong presence of imidazole. That's what it shows. Okay. So again, understand what those tags are. We talked about 2 different tags, right? GST tag and 6 histidine tags. Understand what benefits do they provide. They provide the benefit of what? By tagging your protein, you can purify your fusion protein over affinity column. So you can purify your protein very efficiently. That is the main benefit. Okay. And then we're going to change our topic of discussion. We'll talk about some fundamental calculations that are used in chromatography preparation. Okay, when you understand chromatography system and running conditions, you need to understand these terminologies, and you need to understand some math-based validation method. That's what we're going to talk about. Okay, so when you operate column chromatography in a large scale or small scale, same way, you have to prepare your column. We call that column packing. Okay, so your column should have a defined volume. We call the column volume. Okay, and your resin is introduced to the empty column skid as a slurry. It's a mixture of column resin with some kind of buffer. And then once you pack, meaning once you introduce the column resin slurry, and you need to pack your column, so there's no air pocket in your column present. So you need to compress your resin. Okay, and once it's all compressed and packed, then you need to evaluate how good your column is. Okay, you use a pulse test when you do that. We'll talk about that. Okay, when you do the pulse test, you have to understand how you calculate the HETP value, and then columns asymmetry will be also evaluated. So there are 2 column packing validation methods. One is to calculate HETP value, and second one is by looking at the peak symmetry or asymmetry, you understand how well packed your column is. Okay, and then operational parameters of your column chromatography method. How to scale up your column from the bench scale column you use in the PD group, and how you scale up to a large scale column operation and the manufacturing flow model. Okay, that's the principle. We'll talk about that, and then we'll talk about maybe washing and elution strategy. That's a gradient washing, how you develop the method, and how do you determine the loading capacity of your column and monitoring the process and things like that. Okay, first of all, column packing, like I said, this is a huge column, probably taken a picture from Catalan, and maybe about 1 meter diameter column. Okay, so there's a resin slurry. It's well packed, meaning this metal plate is fully compressed down. So this resin is all packed. Okay, there's no air bubble pocket, anything like that exists. It's all homogeneously well compressed resin. That's what you need. Okay. Then how much of the volume is existing here in this packed column? So you can calculate out. Geometry calculation is what you need. So to define the column volume, that is what? Height times radius square times pi. That's going to give you the volume of this column, right? So that it will be the column volume. So if the height was 20 centimeter, and then if the radius is 15, so 15 square times 20 times pi, which is about 137 milliliter column volume. That's how you can mathematically calculate it out. And slurry calculation, how much slurry can go in there into the column volume. So you need to figure it out. Okay, so you slowly introduce your resin slurry. Okay, using maybe manual introduction, or use a pump. Slowly, continuously, you need to introduce that, and then once it's all settling down, there's a settled volume, and then slurry volume. Okay, so there's a percent slurry volume that needs to be calculated, and it's about 76% if you calculate it, because settled volume is here, and then slurry volume from here to here. So if once it's settled, it becomes denser. That's what it means. Okay. And compression factor, once it settles, then you need to compress your resin down mechanically. I mean, this metal plate should be lowered, right? So how much pressure you need to provide, and how much depth you need to go, you need to figure it out. Settled volume divided by target volume. There may be a target volume, column volume, that is known by the PD group that will be given to you. So you need to press it down until you reach the target volume. Okay, if the difference is settled versus target volume is about 3 centimeter height difference, it's about 1.15 compression factor. That's how you can calculate out what the compression factor is. Okay. Once it's well compressed down, so you need to evaluate if that column is well packed or not. So how would you define a well packed column? Well packed column should have homogeneous distribution of the pressure, and it will guarantee you uniform flow of materials over the column. Okay, it's going to go over the resin uniformly. It's not going to go only one direction or anything like that. There's not going to be any big air pocket that will hinder the flow of the materials, right? So appropriate compression ensures reproducibility between the run. If you are using the same well-packed column, you will have the same result. That's the goal. Okay. These would be considered as not well-packed column, right? If it's overcompressed, okay, and there may be too much pressure in there, or if it's undercompressed, there may be some pockets of air holes there. Neither of them are good. Okay, so how do you validate it? Of course, there's a mathematical calculation that you can use. We call that a pulse test. I'm going to stop right here. I'm going to continue.